Transplant rejection occurs when transplanted tissue is rejected by the recipient's immune system, which destroys the transplanted tissue. Transplant rejection can be lessened by determining the molecular similitude between donor and recipient and by use of immunosuppressant drugs after transplant. The first successful organ transplant, performed in 1954 by Joseph Murray, involved identical twins, and so no rejection was observed. Otherwise, the number of mismatched gene variants, namely alleles, encoding cell surface molecules called major histocompatibility complex, classes I and II, correlate with the rapidity and severity of transplant rejection. In humans, MHC is also called human leukocyte antigen, though cytotoxic cross-match assay can predict rejection mediated by cellular immunity. Genetic expression tests specific to the organ type to be transplanted, for instance AlloMap molecular expression testing, have a high negative predictive value. Transplanting only ABO-compatible grafts helps prevent rejection mediated by humoral immunity. Because very young children recipients, while focus has been on infant heart transplants, the principles generally apply to other forms of solid organ transplantation. The most important factors are that the recipient not have produced isohemagglutinins, and that they have low levels of T-cell independent antigens. UNO's regulations allow for ABOI transplantation in children under 2 years of age if isohemagglutinin titers are 1:4 or below, and if there is no matching ABO recipient. Studies have shown that the period under which a recipient may undergo ABOI transplantation may be prolonged by exposure to non-self A and B antigens. Furthermore, should the recipient require eventual retransplantation, the recipient may receive a new organ of either blood type. Limited success has been achieved in ABO-incompatible heart transplants in adults, though this requires that the adult recipients have low levels of anti-A or anti-B antibodies. Kidney transplantation is more successful, with similar long-term graft survival rates to ABO transplants. Rejection is an adaptive immune response via cellular immunity. Different types of transplanted tissues tend to favor different balances of rejection mechanisms in animals. Exposure to the antigens of a different member of the same or similar species is allostimulation, and the tissue is allogenic. Transplanted organs are often acquired from a cadaver, whose tissues had already sustained ischemia or inflammation, dendritic cells, and present the donor's self-peptides to the recipient's lymphocytes. Lymphocytes include two classes that enact adaptive immunity, also called specific immunity. Lymphocytes of specific immunity T cells, including the subclasses helper T cells and killer T cells, and B cells. The recipient's helper T cells coordinate specific immunity directed at the donor's self-peptides, or at the donor's major histocompatibility complex molecules, or at both. When memory helper T cells CD4 receptors bind to the MHC class II molecules which are expressed on the surfaces of the target cells of the graft tissue, the memory helper T cells T-cell receptors can recognize their target antigen that is presented by the MHC class II molecules. The memory helper T cell subsequently produces clones that, as effector cells, secrete immune signaling molecules in approximately the cytokine balance that had prevailed at the memory helper T cells priming to memorize the antigen. As the priming event in this instance occurred amid inflammation, the immune memory is pro-inflammatory. Transplant rejection is a type 4 migrate to the recipient's lymph nodes and activate T cells that reside there, or an indirect pathway, in which the recipient's dendritic cells can endocytose donor proteins such as donor MHC proteins, and activate T cells in secondary lymphoid tissues. Alloreactive killer T cells, also called cytotoxic T lymphocytes of the killer T cells, recognize their matching epitope and trigger the target cell's programmed cell death by apoptosis, developed through an earlier primary exposure that primed specific immunity to the non-self antigen. A transplant recipient can have specific antibody cross-reacting with the donor tissue upon the transplant event. A secondary exposure. This is typical of minor blood group exposure following allogenic blood transfusion or trauma. During pregnancy, At secondary exposure, these cross-reactive antibody molecules interact with aspects of innate immunity, soluble immune proteins called complement and innate immune cells called phagocytes, which inflames and destroys the transplanted tissue, secreted by an activated B cell, then called plasma cell. An antibody molecule is a soluble immunoglobulin whose basic unit is shaped like the letter Y. The two arms are the Fab regions, while the single stalk is the Fc region. Each of the two tips of Fab region is the paratope, which binds a matching molecular suc.